Biology with Dr. E.
Active transport can be divided into primary active transport or secondary active transport. In primary active transport, energy to do work comes directly from hydrolysis of ATP by transport proteins called pumps. Energy from hydrolysis of ATP causes change in shape of transport protein, which causes a solute such as ions bound to the protein to be pumped across the membrane. An example of such pumps is a sodium-potassium pump or sodium-potassium ATPase on the cell membrane. As already mentioned, the sodium-potassium pump or sodium-potassium ATPase is an example of a primary active transport process. The pump is an enzyme that hydrolyzes ATP to release energy required to move ions across the membrane against their concentration gradient. 3 sodium ions are pumped out while 2 potassium ions are pumped into the cell. This figure shows the role of this pump in 6 steps. In step 1, 3 cytoplasmic sodium ions bind to the pump protein on the cell membrane. In step 2, sodium binding promotes hydrolysis of ATP. This reaction releases energy, which phosphorylates the sodium-potassium pump. In step 3, phosphorylation causes the pump to change shape. The change in shape pushes the sodium out of the cell. In step 4, 2 extracellular potassium ions bind to the sodium-potassium pump. In step 5, the binding of the potassium ions triggers release of the phosphate. Once the pump is dephosphorylated, it resumes its original shape or conformation. In step 6, the pump protein binds ATP and releases potassium into the cytoplasm. The pump's binding sites are now ready to bind sodium ions again, so the cycle can be repeated. The sodium-potassium pump is essential in maintaining the electrochemical gradient across the membrane, which involves both the concentration and electrical charge of ions. This is critical for the function of muscles and nerve tissues. Next, we discuss secondary active transport. In secondary active transport, energy stored in concentration gradient of ions created by primary active transport pumps is used to move substances across the membrane against their concentration gradient. Secondary active transport always moves more than one substance at a time using a co-transport protein. Secondary active transport can be divided into 2 types, symport and antiport. Symporters transport 2 different substances in the same direction. Antiporters transport one substance into the cell while transporting a different substance out of the cell. Some sugars, amino acids, and ions are usually transported into the cell via secondary active transport. This figure illustrates secondary active transport using the ATP-driven sodium-potassium pump as an example. Note that sodium ions are more concentrated outside the cell and potassium ions are more concentrated inside the cell. In the first step, which is a primary active transport process which we've discussed already, the ATP-driven sodium-potassium pump stores energy by creating a steep concentration gradient for sodium entry into the cell. The pump drives sodium ions out of the cell and pumps potassium back into the cell. In the second step, which is a secondary active transport mechanism, sodium ions diffuse back across the membrane through a membrane co-transport protein. This drives glucose against its concentration gradient into the cell. The sodium-glucose symporter loads glucose from the extracellular fluid and moves it across the membrane. Then the sodium-glucose symporter releases the glucose into the cell cytoplasm. Our next topic is vesicular transport. Vesicular transport moves large substances across membranes in structures called vesicles. Because it is an active transport mechanism, it requires energy in the form of ATP. We will focus our attention on 2 main types of vesicular transport, endocytosis and exocytosis. Endocytosis is transport into the cell, and there are three types that we'll talk about: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Exocytosis is transport out of the cell. Endocytosis is transport into the cell and involves formation of protein-coated vesicles. It usually involves receptors and therefore can be a very selective process. So substances being pulled into the cell must be able to bind to their unique receptors. Unfortunately, some pathogens are able to hijack the receptors for transport into the cell. Once a vesicle is pulled into the cell, it may fuse with a lysosome or undergo transcytosis. This figure illustrates 6 steps in endocytosis. In step 1, a protein-coated pit in the cell's plasma membrane ingests a substance. In step 2, a protein-coated vesicle detaches from the pit and into the cell. It loses its protein coat as it moves further into the cell. In step 3, the coat proteins are recycled back to the cell's plasma membrane. In step 4, the vesicle is now an uncoated endocytic vesicle. It fuses with a sorting vesicle called an endosome. In step 5, a transport vesicle may carry some of the membrane components of the fused vesicle back to the cell membrane for recycling. In step 6, the fused vesicle may either A. fuse with the lysosome for digestion of its content, or B. deliver its content to the plasma membrane on the opposite side of the cell. If the vesicle's contents are delivered to the opposite side of the cell, this is known as transcytosis. Next, we review the different types of endocytosis. We start with phagocytosis. Phagocytosis is also referred to as cell eating. In this process, the cell engulfs a large particle by forming projecting pseudopods around the particle. The term pseudopod means false foot. The pseudopods enclose the particle within a membrane sac called a phagosome. The phagosome combines with a lysosome and its contents are digested. The vesicle has receptors capable of binding to microorganisms and solid particles. Next is pinocytosis. Pinocytosis is referred to as cell drinking. The cell takes in drops of extracellular fluid containing solutes. The extracellular fluid enters tiny vesicles. No receptors are used or required in this process, so pinocytosis is non-specific. Pinocytosis is used by some cells to sample their environment. It is also the main way in which nutrients are absorbed in the small intestine. Next is receptor-mediated endocytosis. In this process, extracellular substances bind to specific receptor proteins in the cell's plasma membrane. This process enables the cell to ingest and concentrate specific substances called ligands in protein-coated vesicles. Ligands may simply be released inside the cell, or they may combine with the lysosome to digest their contents. Receptors are recycled back to the plasma membrane in vesicles. Substances taken up by receptor-mediated endocytosis include enzymes, hormones, and some pathogens. The second type of vesicular transport is exocytosis. Exocytosis is a process of ejecting material from the cell. The substances being ejected are usually packaged in secretory vesicles. The vesicles contain proteins called V-snares, while the cell membrane has proteins called T-snares. The two proteins interact and trigger exocytosis. Substances removed from the cell by exocytosis include hormones, neurotransmitters, mucus, and cellular waste. This figure illustrates the 4 key steps in exocytosis. In step 1, a membrane-bound vesicle is shown in the cell's cytoplasm. The vesicle contains molecules to be secreted from the cell. The vesicle surface has membrane proteins called V-snares. This vesicle is migrating with its content to the plasma membrane. In step 2, the secretive vesicle is at the plasma membrane. The proteins on the surface called V-snares bind with membrane proteins called T-snares. In step 3, the vesicle and the plasma membrane fuse, forming a fusion pore that opens into the extracellular fluid. In step 4, the vesicle contents are released to the cell exterior through the fusion pore. Now we turn our attention to membrane potential. As we are aware, the plasma membrane is selectively permeable. The permeability of the plasma membrane results in generation of a membrane potential or a voltage across the membrane. At rest, all cell membranes have electrical potential energy produced by separation of oppositely charged particles across the membrane. The potential typically ranges from minus 50 millivolts to minus 90 millivolts, depending on the cell type. For this reason, all cells are said to be polarized. The minus sign before the voltage indicates that the inside of the cell is negative compared to its outside. The voltage or charge separation exists only at the membrane. While all body cells have membrane potential, it is especially important for nerve cells and muscle cells because they use changes in membrane permeability as a form of communication. So how is the resting membrane potential generated? There are many kinds of ions found inside and outside the cell. However, the resting membrane potential is determined mainly by the concentration gradient of potassium ions and by the differential permeability of the plasma membrane to potassium. The cell cytoplasm contains more potassium ions than the extracellular fluid. The cytoplasm also contains protein anions that are unable to follow potassium through the plasma membrane. As you can see from this diagram, the inner face of the plasma membrane has a negative charge, while the outer face of the plasma membrane has a positive charge. The figure shows 3 key steps in generating the resting membrane potential. In step 1, potassium ions diffuse down their steep concentration gradient out of the cell via leakage channels. This loss of potassium ions results in a negative charge on the inner face of the plasma membrane. In step 2, potassium ions also move into the cell because they are attracted to the negative charge established on the inner face of the plasma membrane. In step 3, when the movement of potassium ions out of the cell equals the movement of potassium ions into the cell, a negative membrane potential of negative 90 millivolts is established. At this point, the concentration gradient promoting the exit of potassium ions exactly opposes the electrical gradient for the entry of potassium into the cell. As we just discussed, diffusion establishes the resting membrane potential. So how is the resting membrane potential maintained? Active transport processes maintain the membrane potential to keep it in a steady state. This requires that the rate of active transport of sodium ions out of the cell is equal to and dependent on the rate of sodium diffusion into the cell. The sodium-potassium pump actively pumps 3 sodium ions out of the cell and pumps back 2 potassium ions into the cell. The sodium-potassium pump maintains both the concentration of ions and charge distribution across the membrane. This makes up the electrochemical gradient across the membrane. The transient opening of gated sodium and potassium ions in the membrane changes the potential from its resting value. This is a normal means of activating neurons and muscles. We will discuss this topic in more details when we get to the nervous system.